Technologies for Pollinator Surveillance in Field Studies and Intelligent Image Recognition, RFID, Rana and DAISY2, presented by Dr. Sara Barlow, Research Ecologist at Red Butte Garden, University of Utah and Associate Faculty, Newcastle University, UK, and Dr. Mark O'Neill, an engineer, software developer and Technical Director of Tumbling Dice Limited, based in Newcastle, UK. Our respective backgrounds have brought together ecological research with novel developments in pollinator surveillance technologies. In so doing, our aim is to contribute to the emerging discipline of e-ecology by developing tools for acquiring, sharing, and understanding data about pollinators to help address urgent knowledge gaps. We have a particular interest in developing pollinator monitoring and tracking tools that are fit for purpose and capable of acquiring large amounts of data in the field. And because this type of digital image data is associated with a bottleneck in data processing, we are also focused on developing a back-end AI system capable of intelligent image recognition. In this talk, we will present 3 novel pollinator monitoring and tracking technologies that we have developed and applied in numerous ecological studies involving plants and pollinators. This work has been undertaken by us over several years in the UK, Europe and Utah. These technologies are, firstly, a new design of prototype RFID tag with wide range for tracking bumblebees. Secondly, an automated visual monitoring system called Rana. And thirdly, an intelligent image recognition system in its second generation of development called DAISY2 that is based on plastic self-organizing maps. Finally, we will conclude with our remarks on the future of e-ecology with particular reference to pollinators. We will begin with an overview of our prototype long-range RFID tags. Commercial RFID, or PIT tags, are commonplace as a means of detecting the comings and goings of managed honeybees and bumblebees at hives or nests and artificial feeding stations. These tiny tags weigh in the region of 3 milligrams, hence they do not unduly affect honeybee behaviours. However, the major limitation of these tags is they have a sub-centimetre detection range, meaning that the tagged bee must pass immediately by an RFID reader to be detected. Whilst this method can be very useful, it does limit experimental design, as these short-range tags are of limited use for tracking the movements of bees, especially wild bees, in response to landscape-scale phenomena, such as pesticide exposure, pests and diseases, and habitat loss and fragmentation, as well as the interactive effects of these stressors. These ecological questions and research requirements apply to honeybees and wild insect pollinators at large. In response, our aim was to develop a prototype RFID tag with a range of at least 1 metre that could be flown by bumblebees, with a view to establishing proof of concept and paving the way for an optimised second generation of tag. Our prototype tags use a Murata strap chip with a custom inductively loaded half-dipole antenna and a 2-watt reader equipped with its own antennae to receive the tag's transmission. The tag is 7 by 2 millimeters plus the coiled antenna and weighs 81 milligrams. We tested the prototype tags in a large glasshouse using a commercial nest of the bumblebee Bombus terrestris. Worker bumblebees weighed in the region of 160 milligrams, hence tags weighed 50% of bees' body weight. We found that bees could take off and fly with the tags attached, and the tags had a maximum detection distance of 1.5 metres. Proof of concept has been successfully established, and with further development, we are likely to improve the system by increasing the detection range further and by reducing tag size and weight to reduce impact on bee behaviour. With further optimisation, including miniaturisation, these tags could be suitable for tracking smaller pollinators such as honeybees. We envisage a production system being used to detect and track bee movement pathways within a designed network of field-deployed low-cost readers and aerials. Shown here is a simplified example of how tags could be applied to mapping honeybee movement pathways in a mixed agricultural landscape to solve problems in bee health and actively assist the delivery of pollinator services. Ultimately, the new tags could provide an effective, low-cost solution to tracking bees in the field, but could also be used in a wide variety of other scientific and commercial applications. The second e-ecology tool we will discuss is an automated visual monitoring system called Rana. Measuring pollinator activity in the field is notoriously difficult to achieve with conventional manual methods that rely on human observations and which can result in data and sampling effort that is lacking in robustness. The solution is to automate the monitoring process with a field-deployable automated visual monitoring system that can vastly increase sampling effort for reduced logistics and labour. This is the purpose of the Rana system. Unlike standard wildlife cameras that use infrared to trigger motion detection of warm-blooded animals, the Rana system uses visual motion detection, or computer vision, involving motion tracking and blob detection to detect focal moving objects such as bees visiting flowers. Rana monitors the live video stream, and when target motion events are detected, the camera is triggered to record and compiles the footage as time-compressed movies of all detected visitations. Typically, a daily 12-hour monitoring period could be condensed into a few minutes. Here is an example of a Rana time-compressed movie capturing foraging visits by pollinators to the rare plant homegrown's milkvetch. Here we see long-tongued anthophora bees, a nectar-robbing wasp, a hawk moth, and a hummingbird. More examples can be found on my YouTube channel. The Rana system uses cheap commercially available hardware, and one Rana unit consists of a digital web camera, a wireless network data logger, such as a Raspberry Pi or Odroid, and a micro SD card to house the software and store data as time-compressed videos. The control web interface is accessed via a wireless connection to a smartphone and allows the user to tune the system remotely, for example, to set the start and stop times of active monitoring and the threshold size of the target object. The smartphone is also able to view the live video stream and download data remotely in the field. A significant advantage of an automated monitoring system like Rana is that camera units can be deployed at multiple sites over continued periods of time to collect large data sets at biologically meaningful temporal and spatial scales. These time-compressed movies are processed manually using video editing software to build large and comprehensive data sets on pollinator visitation rates, pollinator assemblages and their foraging behaviours, and because all visits are time-stamped, data on temporal patterns are also available for analysis. With this method, it is also necessary to refer to field-collected specimens to aid with identifying the insects present in the video. In recent years, we've continued to develop Rana and applied it within ecological research involving plants and pollinators in a variety of ecological contexts, and often at very remote field sites. The technology could, however, be used in a wide variety of studies involving invertebrates, and as a precision agriculture tool, for example, to measure pollinator services by honeybees to crop plants. Most recently, I have used Rana to monitor pollinators of rare and endemic plants in Utah for conservation purposes. This involved a landscape-scale experimental design using 15 Rana units deployed at 10 populations over 2 flowering seasons. Here is an example of a time-compressed movie from that study showing osmia bees and specialist pollen wasps pollinating a rare species of penstemon that is under threat from energy development. Note again the timestamp in the bottom right-hand corner. This study resulted in an extensive data set of 27,000 pollinator visits, captured during a 3,500-hour monitoring period that of course was greatly condensed into time-compressed movies. While the technology enabled a vastly increased sampling effort for reduced logistics and labour compared to conventional manual observations, the major downside of collecting such large volumes of video data is the subsequent bottleneck in data processing and manually translating video observations into metrics. Another consideration is that a lack of appropriate taxonomic knowledge by the analyst will significantly reduce the accuracy of that data extracted.
The solution to this bottleneck is a back-end AI system. In the first instance, this system will filter out frames which contain spurious motion, i.e. due to noise. This is accomplished using DAISY2, a deep learning framework to analyse motion frames detected by Rana. It does this by analysing the input frames of the neural net model built using pre-labelled examples of good and bad motion. If the frame contains good motion, that is a pollinator or pollinators, it is retained, but frames containing spurious, bad motion are discarded. Filtering spurious motion will significantly increase the rate at which Rana data can be analysed, as bad motion, primarily due to wind, can be very significant. In the future, the framework shown here will be further refined for its automated genus and/or species identification, and with the advent of new powerful single-board computers such as the Odroid C4, all of this processing can be done at the edge. And the motion movie which the user sees will be noise filtered with motion sources, in this case pollinators, correctly identified. So, how well does DAISY2 do? In the example shown here, DAISY2 was 100% successful classifying pest and non-pest invertebrate species found on soft fruit. This is a task of comparable complexity to noise filtering, but this result may not be typical. But it is early days. Automated noise filtering has not yet been trialled in field-deployed production systems. When it is, with appropriate training sets and algorithmic refinements, we expect DAISY2 to detect upwards of 80% of the bad motion, and in all likelihood it will perform far better than this. And based on our previous experience, we think that it will be able to identify 70% plus of pollinator motion sources to genus and/or species if examples in the training sets are labelled appropriately. Why DAISY2? It is faster than classical CNN-based deep learning algorithms such as ResNet. The training time is 45 minutes as opposed to upwards of 72 hours, and models can be built and material classified on low-power hardware, that is, low MPCs and single-board computers as opposed to a rack of GPU-equipped blade servers. And lastly, it is capable of fast dynamic learning. Classification models can be extended on the fly. So, what can we say about the future? Well, we envisage a platform which will embed the technologies which we have discussed within an extensible software framework. Data will be acquired in the field directly from a variety of sources including radar, RFID tags, pressure, humidity and temperature sensors, motion and audio. We will also acquire proxy measures for ecosystem function using techniques such as remote sensing. Then, an AI, based in part on DAISY2, will fuse and analyse this data to generate indices of ecosystem functionality. This will produce outputs which inform policy, mitigate ecosystem degradation, and provide the means to restore degraded ecosystems. The emerging discipline of ecology will deliver powerful tools to combat biodiversity loss and climate change.